All right, hello class. So our do now today says how do independent assortment and crossing over account for genetic variation. So pause here and take a couple minutes to try to answer this question based on the POGIL and the information that we got about meiosis from last week. Okay, so when you're done, then start it again. So first we see independent assortment. So independent assortment means that the homologous chromosomes of each pair move independently from one another. So if we look at this example, this has 4 chromosomes or 2 homologous pairs. So we can imagine that in the original individual, there's the red chromosomes, which we could say, just for an example, came from this individual's mother, and the blue chromosomes, which came from this individual's father. So if we look, when they line up at the metaphase plate, in meiosis 1 to separate, it doesn't matter the order in which they line up. So depending on the order in which they line up, so it could be both blues on one side or a blue on one side and red on the other side, you're going to get different results with slightly different information because remember the homologs contain slightly different alleles or slightly different versions of each gene from one another. So where this one might be for blue eyes, this one could be for red eyes, depending on the animal. And where this one might be for brown fur, this one might be for white fur. So it just depends on the animal. So then we see different combinations, okay, depending on how they line up in metaphase plate. So in the end of meiosis 2, that's going to give us slightly different results as far as the information within each of the gametes. So for this, we see that there's not that many, okay? So because there's only 2 chromosome pairs, okay, there's only 2 pairs and there's 2 homologs of each pair, the possible results are just 2 to the second power or 4 different types of gametes. So you see 1 type of gametes, 2 type of gamete, 3, and 4. Now for us, we said that we have 2 homologs, right? But we have 23 pairs. So the possible results are 2 to the 23rd power, which is over 8 million different combinations in the gametes. So that means that every individual has potentially way over 8 million. So it's like 8 million, I think 300,000 in some different gametes. So that creates a lot of possible variation in our offspring. When we look at crossing over, well, crossing over was when these homologs, okay, when they form a tetrad, so when they form that group together, when they potentially cross over one of their little arms, okay, and they exchange that information, so that actually means that part of this arm broke off and switched with the homolog. And this is going to give us slightly different combinations as well, because we can see these letters represent those different genes, okay, on the chromosome. So we could see now we have on one side whatever the capital A is. So if it's brown fur, capital B could be maybe blue eyes for this individual, and maybe capital C is like curly fur. And on the other hand, or on the other side, we've got a new mix. So we still have the capital A and the capital B, but this time we have the lowercase c. So that might be, for example, instead of the curly fur, in combination with these other 2 characteristics, we'll get straight fur. Okay, so we just see this new recombinant DNA chromosome. So let's look at today. So today you're going to do a reading and Cornell notes. So Cornell notes are the 2-column notes. You should be familiar with them. Each of the topics within the reading should be its own big idea in the Cornell notes. And then the supporting ideas should come from the paragraphs, okay, within each subtext or subcategory. At the end of Cornell notes, there is a summary, so make sure you write a summary. And you'll also notice that at the end of the reading, there are 3 analysis questions, so I'll ask that you answer those in your book as well. After that, I'm going to post a video. The video is just showing how the sperm and egg cells come together to form a new individual. And then there will be a Google question on the website, and you need to respond to that Google question. So these are really your 3 main points for today. I was going to do a debrief at the end, so when you're done, when you're done reading, you can come back to the debrief, or you could just keep watching now. But there's a couple things that I wanted to share as we're moving in. Actually, this is just an introduction, so I meant to share this earlier. But the homologous chromosomes, as a reminder, because people are asking lots of questions about them, remember they can contain different alleles. So alleles are just different versions of the same type of gene. So that might be, in this case, if we're talking about flowers, maybe this type of plant has either purple flowers or white flowers. And just so you know, we use different letters to represent those characteristics. So if purple is dominant, then we usually take the P for purple. We make that a capital P representing the dominant characteristic. And then if white is recessive, then we'll use the lowercase p to represent the recessive version of that gene. I put the little underline just because, well, capital P and lowercase p look pretty much identical. So the little underline just helps us separate that this is the lowercase one, okay? So as a reminder, meiosis versus mitosis, some people were saying they were getting confused. So mitosis, remember, makes identical daughter cells. So if this is our original parent cell before synthesis occurs, right, this is after synthesis occurs, of course, because we see the two sides, the two sister chromatids there forming the complete X chromosome. And here we just have the bars. So this is our parent cell. These are our daughter cells. So they're totally identical. So this is mitosis. On the other side, of course, we see this synthesis that occurred. So we're going from our simple bar chromosomes to the X-shaped chromosomes as well. But in the end, we're producing 4 daughter cells that are haploid. So they each have half the number of chromosomes. So if our original cell, its diploid number is 4, so 1, 2, 3, 4 chromosomes, each of these cells will have 1, 2 chromosomes. And we can see here that crossing over did occur. So we see some recombinant chromosomes at the bottom. And just so you know, the crossing over occurs very frequently. So it's not an unusual event. It occurs, I think they believe, in almost all of our daughter cells. Finally, some people are asking about meiosis and mitosis, and this is where the reading that we'll do today will provide more information. But if we look at the process, sorry, not meiosis and mitosis, we're talking about spermatogenesis and oogenesis, so the production of sperm and the production of egg cells, and how they are a little bit different. So spermatogenesis, and the term Genesis should be familiar probably from your religion class. So Genesis refers to creation. So in this case, the spermatogenesis, we are actually creating the sperm cells. And if we look, we start with our one parent cell. Okay, that is diploid. We go through the process of meiosis, and we get these 4 daughter sperm cells. We see that some differentiation needs to occur at the end. So we have our haploid spermatids, but then they need to do some protein production in order to build the flagellum. So all of that does need to occur. And we see that they're, with the exception of the flagellum, fairly small. So they're not carrying a whole bunch of cytoplasm, and they're not carrying a whole bunch of organelles here. They have this highly specialized tail, like we saw before, which has its own mechanisms and contains a lot of mitochondria just for the motion. And then this is the oogenesis. So remember, Genesis is creation. So now we're creating, ova, essentially. So ova are just the eggs. So we start with our diploid cell again, okay? But something really weird happens. So the divisions of the cells are not even. So we're getting in the first division in meiosis 1, a very large cell and a super tiny cell that contains the DNA. And we can see how unbalanced they're becoming. So this cell is like, 6 times the size of this tiny little cell. And this happens in the second division as well. So we're going to end up with 1 giant egg cell, and then 1, 2, 3 smaller cells that just contain the genetic information. So the reason this happens is that we want our main egg cell that's going to be used to have a lot of the cytoplasm, so it's going to provide the cytoplasm and the organelles. It's going to be a rather large cell. And these other little polar bodies that are haploid, they will eventually just disintegrate. So they're never going to become the actual egg cell. So they'll just kind of be reabsorbed within our bodies. So they're there to contain the DNA, right, so that we do get a haploid egg cell, but we want this egg cell actually to supply a lot of the materials to begin the process of division, to have those organelles so that the cell can grow and develop in the case that it is fertilized by a sperm cell. So that's that's the difference. And the sperm cells can't be super huge, because if the sperm cells had the flagellum and then it had like a giant head, then they wouldn't be able to swim well and they would never get where they were going. So these cells are very, very small compared to the egg cell itself. Okay, so get started on the reading, then you'll follow all the steps that are posted on Google Classroom. All right, have a good day.